From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 10, recorded on July 19th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and non-fiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month. And for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Hello, greetings, everyone. Good to be coming to you from Elde Lab Studios here in the Wasatch Mountains.
Is it hot out there?
Yeah, it's a little warm. Summer's here. So we're a bit north of 90 degrees Fahrenheit. I guess that's weighs in around 33 C, approaching that at least.
It's warm.
Yeah, although I'll tell you what, Vincent, I'm wearing a sweatshirt at the moment. The air conditioning, we're still kind of in the genetics building here. We're trying to modulate it. And
it's too cold, huh?
Yeah, exact. Yep.
I figured up in the mountains it'd be a little cooler, but it's not so cool there.
Yeah, if you could keep heading higher and higher,
cooler,
it's off, and so you can escape.
Well, here it's 27 Celsius.
Not too bad.
Nice sunny day with a few puffy clouds in the sky. We had a fierce rainstorm yesterday.
Oh, nice.
But I have been away for 3 weeks, Nels.
Oh, wow. Just getting back from the road?
I got back on Friday, so this is the first week I'm back. I was on vacation with my family.
Oh, very
I
nice.
was in Japan.
Oh, wow. Whereabouts?
We went to Tokyo, Kyoto, Hakone, and an island called Naoshima. Man, I had a great time. I haven't taken such a long vacation in decades.
Wow, good for you.
But 2 of our 3 kids are off to college in the fall. We figured we'd take them on a trip.
Good timing.
And Japan is an awesome place. You ever been there?
Well, just passing through the airport a couple of times.
I'm telling you, it's an amazing country.
Yeah, it's on my list to really dig in.
You definitely have to go. And I figured, well, it's a 12, 13-hour flight. Might as well stay for a couple of weeks. So that was a blast. It was just a blast. And I saw all kinds of cool stuff and wildlife. And so I had fun.
Yeah.
So I'm happy to be back into podcasting. I got 4 podcasts this week lined up.
Jumping in with 2 feet.
Because we got to crank them out. Now that we have sponsors, we have to be regular with our podcasts.
That's right.
The sponsors like regularity.
Fair enough.
And so we are mid-July. The summer is moving quickly, isn't it?
Yeah, it's incredible. So we last crossed paths over at the ASM Microbe a few weeks. That must have been, did you finish up a meeting schedule and then go straight on vacation?
Yeah, so I was at ASM, then I went down to ASV.
Yeah.
I did 2 podcasts at ASM, and one of them you were in the audience, and I asked you to stand up and answer a question.
Yeah, thanks for putting me on the spot.
I figured that you could handle it. Anyone else, I wouldn't have done that to. And then I went to ASV. We did a nice podcast there as well. We had an audience of about 250 people. It was just awesome.
Oh, wow. Yeah, I was really sorry to miss that meeting this year. I was coming off a Gordon conference right before ASM, and then I just got back from one last week. And so I'm kind of had the meetings stacked up. But so ASV was really good this year. I got some positive reports from some of my crew that were there.
Well, I went from ASM on Sunday.
Right.
On Monday, I did TWiEVO at noon. Mm-hmm. And then Tuesday, after a few sessions, I left because I had to get home to see my daughter graduate from high school.
Oh, of course, yeah.
And it took me 12 hours to get home.
Ooh.
So I was flying from Roanoke to Dulles to Newark, and I was sitting in the airport at Roanoke because Dulles was having rainstorms. I just sat there all day.
Oof.
It was just unbelievable. I got home at like midnight.
Yeah, ouch. I snuck in a quick, between all these meetings, we had a quick family reunion up in Minnesota, and we were flying from Salt Lake to Minneapolis, and another rainstorm popped up, and so our plane circled a few times, decided we didn't have enough gas, and so we ended up landing in Sioux Falls, South Dakota. Added a few hours onto the itinerary unexpectedly there.
Yeah, you figure summer flying should be clear, not like the winter, but rainstorms can wreak havoc, so...
Yeah, exactly.
It's a big problem. So next year, ASV is in Wisconsin, Madison.
Yeah, that's always fun. I really like it up there on that campus. It's a good venue.
And then in a couple of years, they're going to be back in Minneapolis, so you might want to...
Oh, yeah, definitely get that on the menu. Were you at the last Minneapolis
version?
I think
I was.
Yeah, me too. That was, speaking of summer weather.
That was hot.
Oh, my goodness. It was like being in a jungle. The humidity and the heat really kind of conspired against us there.
Yeah, it was hot, but I enjoyed Minneapolis, and I'm looking forward to going back.
Yeah, good town. And I was, I was around the University of Minnesota campus a bit, catching up with some friends.
Mm-hmm.
We were taking advantage of all these new bicycle trails and to one of the big microbreweries there, the Surly Brewery, which is a really nice place. So there's a good feel to it. And maybe we should cook up a TWiEVO episode at the brewery. I'll see. I might put out some
that would be
fillers on that. Yeah,
you bet.
See if we could get some cross pollination.
Good.
So yeah.
All right. What about Stan Falco?
Oh yeah. So I was kind of a little note here. So I was just this meeting last week. This was Gordon Conference up in New Hampshire. We had pretty decent weather. Actually quite beautiful in the White Mountains. Up at the Waterville Resort.
Mm-hmm.
And the topic of the meeting was microbial toxins and pathogenesis. And it was really fun to just see all this beautiful biology, how these microbes just, take over cells and manipulate these processes to their advantage. And one of the quotes that kind of stuck with me from one of the speakers was quoting Stan Falco, and his quote that the toxins can serve as tutors. This idea that just by following what the bacterial functions are doing can teach us about our cells. And so, I like that idea. And if anything, would like to extend it to the toxins both as tutors and sculptors, that they shape the evolution, not only sort of teach us about how our cells have processes work, but have shaped those processes as well.
Stan is always good for good quotes.
Yeah, I agree. Definitely.
Stan Falco. Well, that's great.
Yeah. And so, I think... I was coming in, I gave a talk that felt like it was well received by the microbiology crowd. And it kind of reminded me of how, when, this is the first time I was at that meeting. And, we kind of, I think still as communities, whether it's virology, microbiology of bacteria, cell biologists, evolutionary biologists, we still kind of have some cultural boundaries.
Yeah, sure.
And so that was, I think one of the fun things about this was to try to break that down a little bit, and some of the great conversations that emerged where, if we can think about evolution the way we've been trying to talk about it and discussing it here on TWiEVO, where it's not just sort of these dried out ideas from the past, but that really experimental approaches kind of integrate and tie and weave through the work, that's sort of a fun way forward. And so that turned out to be a fun meeting.
Yeah, TWiEVO is a good model for that. Because we cover a lot of things because of the evolution, right? It's a broad subject, not just bacteria or viruses. So
yeah, it gives us some nice latitude to cover ground. And some of those, I bet some of these microbial stories, and bacteria in particular, will start to bubble up a bit more as we continue on.
I think meetings that are cross-disciplinary are particularly interesting, because you always know the people in your field, and you get together, you have a poliovirus meeting,
Yeah, right. Yep. Yeah, and this idea, so sometimes I go to these meetings, and the danger is that I get a little bit pigeonholed, like as the evolution guy who kind of shows up, and and that's fine, fair enough. But I really, what I think the promise of moving forward is that we can just kind of return to the idea that we're all scientists or biologists, and then kind of let all of the different topics kind of filter around us and see how that kind of emerges in new ways. Yeah,
you don't want to be known as Chuck Elde, right?
That's right. That's right.
The evolution guy.
Exactly.
All right. Well, today we have a paper, don't we?
We do. Let's see here. So we are moving on to this story. Has been has been out for a while, and after we had a great episode last time with Jim Bull, talking about coming in as our guest and talking about some of his ideas modeling gene drive, we kind of can return to this now. So this is kind of actually harkens back, I would say, to a historical example that's kind of trotted out there as a great illustration of evolution in action. This is the famed peppered moths in Britain, in the UK. So do you remember hearing about this story?
Oh, absolutely. This is a classic, right?
Yeah.
Everybody's heard of this. I mean, they teach it in grammar school, probably, right?
Yeah.
Well, some people don't. You teach it other people.
Sure. No, I certainly remember it from very early. And so it's this idea of industrial melanization that during the Industrial Revolution, we're talking about kind of the 1800s, like early to mid 1800s, when a lot of coal was being burned. What happened? There are some kind of detrimental side effects. So some of those being that all of the smoke and soot started to kill lichens on trees. This darkened the bark, and then you have these peppered moths that are just hanging out in the forests. And up until the Industrial Revolution, the idea goes that they were perfectly camouflaged with this sort of peppery or molted coloring, and they just blend right into the lichen. We'll put up some images on the TWiEVO site because it really is striking how these things can kind of just disappear on the bark of trees. And then as soon as that sort of camouflage goes away, in the sense that the bark is darkened by all the coal, then the next part of the idea is that the birds around there that would eat these moths sort of have a field day because they're just completely exposed. And so then what happens, or the hypothesis, is that as darker morphs or variants of the peppered moths showed up, this is all of a sudden a massive advantage for them, because now these dark moths that can be almost completely black in color will blend into the new environment, the darkened tree bark. Right. And so there are these, great stories then of how fast basically the entire population of moths could shift based on the pressure from the birds as predators, and then this really quick and stark environmental change, which was the loss of camouflage through the burning of all of that coal.
And did this happen throughout the UK or just in certain places? Do you know?
Yeah, it's a good question. I think it was somewhat. The ideas was observed even more sort of noticeably near the big cities.
Yeah, that's right.
Since that's where all
being burned. Yeah,
correct. And that actually, I think was part of the early hypothesis, was that it was so evident there and not so much so in the countryside. Yeah, and so I think that fed into it. It turns out it still was a really controversial idea, and part of that controversy cropped up, years later. I think some of the early experiments on trying to actually measure bird predation of dark versus peppered moths on different substrates. In this case, people were setting up kind of artificial natural environments, so to speak, and see whether birds had an advantage when you mismatched the camouflage to the environment. And I think some of those early experiments were sort of consistent with the idea, but then those were challenged by other scientists who said, wait a minute, in part of setting up this artificial system, you've trained the birds to have a specific, ability to hunt moths under certain conditions. And so it really ended up kind of going out the window, I would say, for a while. In fact, I think part of the, sort of what makes this a classic example or sort of famous is there's also then this collision and controversy with sort of the religious corners that sometimes argue with the idea that evolution happens at all, intelligent design in particular. And so this was sort of held up by that crowd as examples of how these evolutionary biologists are sort of scam artists or fraudsters. And so it was sort of a rough, sort of era for the peppered moth hypothesis for many years. And then, but what happened even just in the last kind of decade or so, there was a fellow called Michael Majerus, and he kind of set out to revisit the kind of all the way back, the historical evidence, the experiments kind of in the 50s and 60s. And then he ended up organizing a really big, large-scale experiment, I think they released something like 5,000 moths, tried to correct for some of the controversial points that were brought up or the potential artifacts that were put out there. And it turns out he actually passed away before the experiment was completely finished. This is the early 2000s.
It's a bummer, huh?
Yeah. Yep.
So never got finished then?
Yeah. Well, so actually what happened was a few of his colleagues sort of picked up on the work. And one of the silver linings here is they were able to kind of draw together the loose ends a little bit, and then published about 3 or 4 years ago a paper that sort of covered all this. We'll put a link to this on the TWiEVO website, and it's kind of interesting. So there's a couple fun TWiEVO connections here. So the first is one of the authors on this paper that's in Biology Letters, 2012, I believe. And the title of the paper is Selective Bird Predation on the Peppered Moth, the Last Experiment of Michael Majerus. So they are able to kind of honor that work and then also sort of put to rest, actually, some of that controversy. So the evidence was considered pretty convincing that this idea of predation in the changing environment really did sort of hold up to some of the more original ideas.
So what did they do? They actually released moths and counted white versus black ones, how they were predated by birds. Is that right?
Yeah, exactly. So, and there's a few, it gets a little more complex than that. And, in order to do this, so they released, I think it was 4,864 moths, and it took 6 years to do the experiment.
How the hell do you go count them? Don't they fly off and you never see them again?
That's right. And so that's right. So that was the part of the trick and that was, I think, getting around some of those criticisms was that they wanted to make it as natural as possible, right? So some of the earlier controversy was that, you're gluing these moths to tree branches, and how realistic is that? And so this is kind of an interesting case of combining, I would say, experimental biology with evolutionary biology, where you have these compromises between how do you do a controlled experiment as much as you can, but keep all of the variables of a natural setting. And so it's kind of an interesting example of some of the tension, I think, between that, and worth reading up on this kind of fascinating study, long-term study, and then also kind of ties in all of this history as well.
So this release experiment the aim was to figure out what the selection was, right?
Correct. So so it was very clear, just from simple observations, that the populations of these peppered moths have certainly fluctuated in the frequency of the different color morphs. But whether or not that was just, your null hypothesis could be that this is sort of a neutral process of drift, just by chance that's happening. Yeah. And so what you're trying to then discern is whether there's actually natural selection acting on top of that. And that's where the birds come in, and these predators and the idea of camouflage.
Now, my understanding is that as the UK cleaned up its act and got rid of the pollution, that the black moths kind of decreased in numbers, and we went back to the peppered moths, right? Is that correct?
Yeah, exactly. And it's really, I think it's striking. And so that's, kind of this delay. I think it's not even until something like the 70s when the populations really make a drastic shift from, say, 90% or something around that level of the darker morphs, and then back to, 95% of the peppered morphs since then. And so now these things, I think my understanding is that they're pretty rare, that you'll find the darker colored moths.
So for the experiments we're going to talk about today, and for some of them that have been published, then you can breed the black ones in the lab, obviously, right?
That's right. And actually, you can still find them. And so some of the, we'll get into sort of the collecting. They actually have done some more recent collecting, and then include some of the, you can freeze them. Once you collect them, you can freeze them down if you're just sequencing their genome or genotyping them in some way. So
why would it that if you have all these black moths and then you remove the selection for them, why wouldn't they just stay black? There must be some negative property of being black moth, right?
Yeah, correct. And so it's actually, it's not just sort of that you would drift back or something like that, but then, so as the lichen grows back on the trees and the tree bark lightens up, the idea is counter selection is that now if you're a black moth, you're sticking out like a sore thumb. Exactly. So yeah, so it's both. It's a really beautiful kind of clean reversal where there's an advantage, a strong advantage in both directions, depending on the substrate, which is this kind of crazy industrial pollution that our species is responsible for in this case.
So this whole field of studying things that happen out in nature, they call ecological genetics. Is that right? Yeah,
I think that's one of the common ways of capturing it. And yeah, I mean, you can think about this as sort of genetics in the wild, right? So,
yeah,
you know, when we've talked some about the rise of Drosophila as a model system,
yeah,
where that's in a lot of ways, that's certainly not ecological genetics.
Yeah,
where it's
nor were the pigeons, right?
Yeah, good point. Yeah, where you have all this artificial selection,
but the butterflies were, right?
Yeah, exactly. And so I think that's a really nice example of ecological genetics. And then this idea that now that we're in the kind of genome era or post-genome era, that there are these new tools to go from the observations and sort of all of the complexity of the wild ecological habitats and then try to capture that in new ways. Sort of what are the key genetic points of tension or turning points for how things are adapting and how the fitness of organisms is affected in these complex environments, what's adaptive, what's not. And so to be able to start to dissect some of that complexity in new and exciting ways.
Would you consider some of your work ecological genetics?
Yeah, that's a good question. I mean, so the ecology of the host microbe interface, right? I think it does start to get at that a little bit because, you know, the ecosystem is pretty limited. It's the host. But we are starting to wrestle with a lot of the complexity that can emerge from that. And so, you know, I think in my group, we're so far have taken a pretty traditional reductionist approach in the sense that we're always looking for sort of that single amino acid substitution or that functional variant, whatever the mechanism is. But if we look at things like sort of what's happening in the area of microbiome research, I think that has started really as a acknowledgement of the biological complexity that you can find in, say, the gut or other locations, even on our bodies. And so, and I think we've even seen an echo of this sort of development of the field where the first era of microbiome work, and we're still somewhat in this era, is to really just catalog or get a sense of how complex these things are, to count, you know, you see a lot of these sort of surveys of what is the diversity, what are the species, the strains or whatever, without a whole lot of functional sort of interpretation or experimentation. And now I think that field is sort of embarking on this next exciting era, which is to then actually go in and sort of experimentally manipulate these systems and ask how these things change as well. So yeah, I think about it that way.
They make the point that this whole field of ecological genetics has been revitalized by next generation sequencing, because they say, you know, we reached a point where we couldn't figure out what was going on by just crossing, because you get to, I don't know, 400 kb region or a 200 kb region. Right. And okay, we can't get any smaller. But now sequencing opens it up, right? And this is the theme of TWiEVO, right?
Yeah, exactly. I think so. And and it's, you know, these new tools sort of give you a new way to go farther, to sort of take a lot of that complexity and look at it in a new way, bringing sort of the experimental approaches out to the field. So like we have this rise of the Oxford Nanopore sequencing platform, where the sequencers themselves could be the size of, harmonica. Basically, you plug this into the USB of your port on your laptop and you're sequencing genomes. People are doing this in the field to monitor Ebola evolution kind of in real time. And so I think it really is an exciting, it certainly has invigorated the field. That's for sure.
Yeah.
One example, another example, I think, where we could kind of look at this is Rich Lenski's really interesting work, the long-term E. coli evolution experiments, where this is more laboratory than ecological, certainly, but he, basically was just for the last 30 or so years carefully pipetting a single starting E. coli strain in 12 replicates from one flask to the next. counting up in the thousands of generations now. I think they're past 60,000 generations. And a lot of these interesting observations of adaptations just sort of sat in the -80 freezer until genomics kind of caught up, and you could sequence these genomes and start to tie the phenotypes, to the genotypes and get at the genetic basis in some of these cases. So kind of echoing all over the place in really cool ways.
So the title, this is a Nature letter. The title is "The Industrial Melanism Mutation in British Peppered Moths is a Transposable Element." And the first author is Arjen van 't Hoff. The last author is Illik Sakari. Do you know any of these individuals? They're from the UK.
Yeah, University of Liverpool. I haven't come across them. The senior author, Illik Sakari, is also an author on that biology letters paper that we just spoke about as well. And so I think he's had a hand in sort of doing both the setting up the experiments kind of in the ecological settings, that Majerus experiment, and being involved in that all the way through now to the genetics here. And then, as we'll discuss, trying to get at some of the mechanism of what was going on. And so
up until this paper, there's been some work done. I'm going to ask you to fill us in on that, but the terms are interesting. So the peppered moth is Biston betularia, so it's a very specific kind of moth.
Yeah, that's the genus and species. That's right.
And then the pale, the whitish form is called the typica form. And the black is the Carbonaria, which made me hungry because it makes me think of
Carbonara?
Carbonara, which I love.
Man, I'm getting hungry now too that you mentioned it. But
and they say the Carbonaria locus has been localized to a 200 kb region. So how would they do that? Basically, can you tell us?
Yeah, absolutely. So this is traditional so-called linkage mapping or genetic mapping, and I think we've talked about this a bit already. It actually takes us back in some ways to the fly rooms at Columbia in your neighborhood there, to the days of Thomas Hunt Morgan and Sturtevant and all those others. And so they're kind of the first to do this in kind of almost industrial scale of basically linking a phenotypic trait. In this case, we're talking obviously about the color or the patterning of these moths to locations or markers on chromosomes. And so those markers can be lots of different things. In some cases, those were phenotypes themselves. Yeah. The red-colored eyes of Drosophila, these classic markers. Yep. More recently, these markers are generally molecular markers, so certain polymorphisms, maybe a restriction enzyme site, or even just some known genetic variation. And then the idea of linking things together in sort of close approximation is that you can estimate that by doing crosses and then seeing the action or inferring the action of recombination. So if two things are farther away from each other, a genotype and a phenotype, for example, they will be constantly unlinked or sort of not associated with each other because recombination will break them up, just the frequency of recombination. Whereas if something is really tightly genetically linked, then it'll be very rare that a recombination event will break those up. And so they'll always kind of co-segregate together or be linked. And so that's what had happened with this Carbonaria, not to be mistaken with the lunch Carbonara locus. Just by doing sort of this traditional mapping, they were able to localize it to something like a 200 or 400 kb region.
Yeah, I misspoke. They got it previously to 400 kb.
Okay.
And then in this paper, they narrowed it down even more. Yeah.
Yeah, exactly. So, and just to kind of maybe highlight some of the tools that were missing. So, in fact, before they started this experiment, there wasn't a genome scale analysis, at least that I know of, done on Biston betularia, the peppered moth. But the closest one is the silkworm moth, Bombyx mori, which is kind of a famous guy. I'm sure you've heard of that.
Yeah, sure.
Yep, comes up all the time. So, obviously for the silkworm, and then also it's a genetic system in its own right. Yeah. So that they're able to use the genome that I think has been out now it was one of the early ones. I think it was more than 10 years ago, and then sort of overlay the sequences that they got back from doing peppered moth genomes, and in fact, in this era, you end up doing so much. It's so easy to get so much sequence kind of so fast that they ended up doing genome scale, level sequencing on this species. But I think it actually raises a bigger point here, that one that I've been meaning to kind of talk about a bit on TWiEVO, is that we kind of throw around this idea that a species or an organism has had its genome sequenced, or its genome is done, and in fact, what that can mean, I think, is really wildly different things. So actually getting the raw data, that is sequencing every base in a genome, to some, and doing that repeatedly to some depth of coverage, as it's commonly referred to, that's actually pretty straightforward. What's really difficult, though, the heavy lifting for genomics, for quote unquote doing a genome or finishing a genome, is all of the annotation, is to take all of those raw sequence reads to actually, first of all, put them together as contiguous regions, as so-called contigs, and then are onto scaffolds, even larger pieces, and then all together as an entire chromosome. This is rarely done because there are so many, just sort of complex regions that don't make sense, where even though you've sequenced a lot, you can't quite put it all together. Mm-hmm. Mm-hmm. And then from there, one of the things that we really want to know about are, well, where are the genes, or what are the sort of functional elements around that? And that takes a lot of heavy lifting, or, in the best case scenario, experimental evidence. And so just sequencing a genome and having a useful genome sequence are two vastly different things.
Yeah, for sure. So another thing maybe you could go over with us is they do a lot of crossing, just use larger population samples and better markers. So they get it down to 100 kb, and there are 13 genes and 2 microRNAs, and they have to narrow it down even further. And they mentioned this problem of the rapid spread of the Carbonaria. Gave rise to strong linkage disequilibrium, so that many sequence variants can be associated with the phenotype. And this poses a challenge for saying which gene is involved. So what does that mean?
Yeah. So this is really interesting. So first of all, I think the before this paper, when they were in that larger region, I think that, like you're saying, had 13 different genes in it, all kinds of variations. Really be at that scale or that resolution, really difficult to put your finger on, okay, this is why, this is linked to the difference in patterns or colors between the moths, the Typica versus the Carbonaria. And so that, first of all, mapping to one gene is great because you now have like a really strong candidate. But then again, as you're pointing out, you're not out of the woods yet. And so in this case, you have this idea of linkage disequilibrium. And just to define that a little bit, it's kind of an enigmatic phrase. It's not sort of intuitive what linkage disequilibrium. I always have to go back and refreshing myself on it. So this is basically in broad strokes. This is the association of genetic variants in non-random patterns. And so this, first of all, kind of opens up the question of, well, what is a random pattern of gene variation? And I guess what you would think of there is that just this idea of mutations, whatever they are, point mutations, other mechanisms of mutation, just sprinkling kind of randomly throughout the genome with no rhyme or reason, right? But then real life is never kind of that simple. And randomness breaks down. So for example, let's say there's a super beneficial mutation that lands somewhere in a genome, and then natural selection acts on that. That's what that's going to do, is basically change the frequency of that mutation. And in fact, a lot of the mutations just next door, because it will, recombination, again, this idea of linkage, takes a while to sort of disconnect that beneficial mutation from sort of the random ones around it. And so that would be an example of where you see a number of genetic variants in a non-random pattern, is that you have these things that, because it's happened so recently and selection has acted on it so strongly, you now have this sort of cohort of variants that are moving together. One or a few of them are the ones that are selected on, and the other ones are just kind of coming along for the ride, these so-called hitchhiking mutations. Right. Yeah. And so in this case, even though they've narrowed it to one gene, they describe something like 87 genetic differences in that variable. And so that's really the question, or the problem, is then how do you go from those 87 down to the 1 or 2 or that actually might be the functionally consequential variant here, the one that turns the moth somehow from the peppered color to the very dark carbon color.
So, all right. So again, I misspoke. So basically, in this paper, they narrowed down the region responsible for the black pigment to 100 kb, and then they say this is basically one gene within this. It's within the span of one gene.
Yeah.
And now
exactly.
the problem is what mutation or variation in this gene is responsible for the phenotype. And as you said, there's so many random mutations that they have to look at lots and lots of Typica and Carbonaria isolates to see which are present in most of them, right?
Yeah, exactly. And so this is pretty cool, actually. So, and the gene is called cortex. It's been a little bit of function has been defined in Drosophila, which has an orthologous gene copy, and we'll get to the function in a bit. But just to stay on this question, and how do you narrow down those 87 mutations? And so here, what they did is they grabbed a bunch of the Typica individuals, and they just sequenced out this region, that 100 kb region, right? And then what they're able to do is say, okay, or the assumption they make is that if this mutation is the same in every Typica case that they look at, as in the Carbonaria, now I'm going to say Carbonaria, Carbonaria form, then they can throw that out because it won't be caught. Yeah, and actually, if you look in the paper, I think it's their first supplemental figure, maybe it's supplemental figure 2. They just show a nice sort of example of that for a little. This is supplemental figure 1B. They show just a little bit of a stretch of the nucleic acids and which ones they would disregard because they're identical between them, or the ones that they would keep. Because there are actual differences between them. Yeah. If that makes sense. Yep. So that got them, they were really able to narrow down, just to a few different mutations, I think a handful, maybe 2 or 3. And there was still at least one case, the way they tell the story, where they couldn't distinguish. And so they went back and sequenced even more of the Typica forms until they found this really rare variant that matched the Carbonaria. And so they could throw that one out. And then what they were left with was actually not just a single point mutation, but this big 20 kilobase insertion by a transposable element, which was the most dramatic difference between the Carbonaria and the Typica genomes in that region.
How would you know it's a transposable element? What are the signatures?
Yeah, so in this case, you're looking, there are some definite signatures here. So this ends up being a DNA transposon. It's a type 2. So this thing is defined. There are sequences that, identify these transposons, and these are based on the genes that they encode. This guy does a cut and paste mechanism. So it will actually cut out of a genomic region, land in another one, and then be able to propagate or move around chromosomes as a kind of in this massive category of selfish genetic elements that just litter many genomes, including moth genomes, as we're seeing in this case. And so, but your question is a good one. I mean, it's not like a complete slam dunk, because a lot of times you'll have sort of these hints of the identity of the transposable element or the selfish genetic element, but sort of they're also picking up neighboring regions. There's a lot of repetitive DNA. And so you definitely have to kind of dig in and really analyze those sequences to define what they are.
Nels, I got a book from ASM a couple months ago.
What's that?
Called Mobile DNA.
Ah, perfect.
And it's about, 6 inches thick. It must weigh over 20 pounds, and it's 1,000 pages or more. And it's everything everyone knows about mobile DNA. It's just amazing. It's an incredible field. Steve Goff tells me that there's this wonderful meeting. I think it's out your way.
Is that right?
Yeah, on mobile DNA. And it's really cool stuff, including this. And he came in today, he said, what do you think would be a good satellite meeting for ASV? And I said, they should have one on mobile DNA. It would be really cool.
Yeah, great idea.
It's a great field. And as you'll see from this, that these DNAs jumping around do good things sometimes.
Totally agree. And so this is, I think mobile DNA has been recognized for a long time and has made connections all the way through so much interesting biology for so long. Yeah. Some of the deep ties to virology. Now, as we're doing more and more of this sort of genomics meets evolution meets biology, this isn't the only time when a mobile element has shown up and sort of, just as it was hopping around or mobilizing through genomes, actually had this sort of beneficial side effect. This is a interesting and emerging theme that we're seeing more and more. So
genomes of living things are full of them. Thousands and thousands of copies, and sometimes they have bad effects. They go in front of a gene, or they go into it and mess it up. So there's some human diseases that are associated with mobile DNA, but certainly there are many examples of beneficial consequences of jumping around as well.
Yeah, I agree. And in fact, I think it's we're really comfortable thinking about the point mutation as a mechanism of evolution, right? So you just change one base pair. But I think about these mobile elements as sort of another mechanism of generating diversity, the same way, where what you would predict is kind of what you're saying, right? That you'll have some deleterious ones that might have disease effects, cause cancer in some cases. There are increasing observations of that. Many that will be sort of very mildly deleterious or sort of neutral, near neutral, which will just sort of hop around and just do their business. And then these other, occasions where we might see something beneficial, as in this case.
Now they have some black moths that don't have this big piece of DNA, right?
Yeah, that's right. These are maybe the exceptions that prove the rule. I don't know exactly. So I think they note 5 of their Carbonaria morphs that actually don't have that transposon. And then they go back. It turns out there's a little more complexity to the story. There's a third guy. So we've already been talking about Typica and Carbonaria. There's a third one. Do you remember offhand? Insularia, which is sort of an intermediate phenotype, somewhere between the peppered and the very dark. And so what they think is that these might just be extreme cases of that insularia, where there's sort of another genetic pathway to these things having a similar morphology, but they don't go much deeper than that.
So 105 out of 110 black moths had this, they call it the Carbonaria mutation, which is really the 20 kb transposon.
Yeah, exactly. Whereas I think it's 0 out of 280 some odd Typicas don't have it. So pretty, I think strong evidence for a genetic association or a linkage here. Pretty hard to beat that, actually.
That's pretty good.
Yeah. And then what's really cool is now that they have kind of put their thumb on that, you can start to look back and say, okay, I mean, the other thing is the idea is that this thing emerged pretty recently, at least in high frequencies. And so they can start to look back and try to retrace its evolution just by doing some population genetics. And so the first, I think, this is just sort of in some way a side note, but they can ask how many copies of this are there actually in these moth genomes. I think it turns out to be something like only 250 copies. This exact, if you just blast search for this sort of type of transposon, there's no record of it in any GenBank file. And so they can make a case that this is like it's sort of a new family, or it's a family specific to this species and in this particular form. And that's relatively rare. I mean, in many cases, these transposons are there's thousands of them, and they can occupy easily as much or more space than the coding regions in a genome.
Sure.
And so it's kind of this interesting side note that this is a really rare thing, and you have these kind of rare events piling up that can have these really big consequences potentially as it plays out in the actual phenotype and then the predation and in the natural selection.
Right.
Yeah.
Can they go back very far? I mean, I don't know if they have museum samples of these moths, and see how old this is. They can.
Yeah, it's a great question. I don't know the answer, but you can imagine sort of a parallel here with all of the kind of like the Neanderthal and the ancient genomes that we've been talking about some on TWiEVO. You could do perhaps a similar look at the moths. So they don't mention that. What they do something a little bit different, is that now that they have kind of mapped out all of these cases and looked at it in sort of hundreds of individuals, then they can use population genetic measures they're inferring population sizes, recombination rates, but then they can ask actually, just by looking at individuals and what's going on in that sort of 400 kb region nearby, how much recombination has there been, and if that's playing out sort of as the estimates or models would have, they can try to put a date using statistical methods on when that DNA transposon, when it moved to that location. And so they have, in their estimate, they show some maximum likelihood methods that give it sort of this landing point that they think the hop might have happened in the year 1820 or in and around there with some statistical confidence. And that is kind of interesting because it loosely matches right with when the Industrial Revolution was happening. And I think, it's a little bit of a lag time. So the real kind of deforestation or effects on the trees might have been happening 30 or 40 years later.
Yeah.
But then the hypothesis would be that jump happened and was just kind of at very low frequency in the population. And then it just really exploded as soon as the environment changed. It was sort of poised to take off. It's kind of an interesting idea.
It's interesting. They say that the first sighting of Carbonaria is 1848 in Manchester, which of course is really where the Industrial Revolution started in the UK.
Yeah, there you go. So maybe that's
makes perfect sense, right?
Epicenter. I mean, getting back to your earlier question, I don't know what kind of resolution you have, but if there's a possibility of convergent evolution, that would be that this would independently happen in different ways if you were sampling around other cities. But I just don't know the history well enough. That certainly happens in other biological settings where populations sort of, if they're separated, or even within the population, you can have independent mutations that sort of give you the same outcomes, and then you can kind of select on them at the same time. But not clear that that's happening in this case. Certainly, at least the evidence that they show here, it's all this like one or few early events that was this big hop of the TE that somehow right into or the region of this cortex gene.
So one of the things they say, maybe you can shed some light on, they say the tenure of the carb TE, so that's the name for this Carbonaria transposable element, has been transient, having declined from 99% to less than 5% in its industrial heartland since 1970. Okay, so that goes with earlier discussion we have of, we've cleaned up the soot and we're going back to the peppered moths. But as they say, it has nevertheless left a substantial trace of its former abundance in the form of ancestral Carbonaria haplotype blocks introgressed into Typica and Insularia haplotypes. And maybe you could explain. So is this saying that today, if you look in Typica and Insularia, you can find this carb TE?
That's right. And so then the question becomes, well, where is it, right? Is it, so the hypothesis would be that if it landed in this very specific area, and we'll talk about sort of that gene in a few minutes, that will have this outcome where you'll switch colors. But if for the 200 or so copies that are just sort of circulating around in the genome, I think what they're saying is that you can still see that, traces of that, basically, right? Yep. And because this wasn't seen before, they actually, I don't know, I'm not actually, to be honest, I'm not super crazy about that name for the TE. I mean, the family is in the hundreds, and they're naming it after this one impact that they're proposing. But maybe there's 10 other hops that had other really cool evolutionary outcomes, and then we're kind of limited on that name. I mean, that's fine.
Interesting. So, but it just if we keep looking at this in the coming years, it would be interesting to know if we slowly lose these other carb TEs, or if they stick around, maybe they have some other function. Who knows, right?
Yeah, or they're just, like a selfish gene will just sort of propagate or hang on by its fingernails in some way as well.
Curious. Okay.
Yeah, it's really interesting.
So the TE integrated is just upstream of this cortex gene, is that right?
Yeah, so then now we're starting to transition from, they did a pretty, and at least in my estimation, a pretty convincing job of linking the phenotype to the genotype, which was, like you're saying, the transposable element landing right near the coding region or right around this cortex gene. But then the next question becomes, well, what did that really do from sort of a molecular standpoint? Did that impact the function of cortex? So that could happen. You see a lot of cases where these transposable elements might disrupt a coding region. It might break the gene, knock it out, in essence. People experimentalists use this all the time. The P elements of Drosophila are used to make knockouts of Drosophila. In other cases, what can happen is it doesn't actually interrupt or change the coding potential, but it changes the regulation of the gene. And in this case, that was there just by looking at sort of the architecture, the structure around the exons and the introns and upstream of the gene. That was their idea, that this guy probably, when it landed, might have altered the transcription. And so that's sort of the genetic phenotype they started to look for, was could they see differences in the expression of this cortex gene?
Yeah.
So we should probably say, so what is the cortex gene? So not a lot was known about it. It's thought to be a gene involved in cell cycle regulation from very early in meiosis, actually. And so that little bit of work, functional work, just by, I think in this case it's Drosophila developmental biologists, doesn't cast a whole lot of light on the idea of, well, why would this turn a peppered moth into a really dark colored moth? But at least it gives them a foothold to go from. And so they end up doing, looking at the expression of this transcript at different stages of development, and basically comparing that in the peppered morphs versus the Carbonaria morphs, Typica versus Carbonaria. And they do see a slight, but I would say reproducible, at least if you look at the data. And this is in figure 3 now. In the developing wings. So they're basically looking at the larval stages, mostly though at the what they call the prepupa stage, and then asking how much cortex transcript is there present in these, in this case, the imaginal discs, these sort of early developing structures that will then become, in the adult flies, or sorry, the adult moths, will become the wings and some of the other legs and other structures like that. And so what they see is that there's greater, I think they kind of narrow it down in the Carbonaria morph to a pre-pupa stage at day 4, where they actually see a lot more, just consistently more, let's just say, expression of the cortex gene.
Yeah, it's, I mean, the numbers are pretty low here.
Yeah, that's right. Exactly. It's a pretty subtle.
I mean, the y-axis goes from 0.005, which is relative expression, to 0.01, like twofold maybe, right?
Yeah, exactly. It's pretty slight. And then they do show, they can compare the different genotypes. So if you have 2 copies, if you're a homozygote for Carbonaria, if you're a heterozygote, so you will have 1 copy of the transposable element at the cortex gene and 1 copy without the Typica genotype, and then or no copies, so you're just the Typica allele of cortex.
Yeah.
And in all of those cases, they do see, a shift that correlates with whether you have 1 or 2 copies in the expression going up. Again, very subtle. As you're pointing out, Vincent, this is just like a twofold change at a very narrow window of development. So maybe I don't know how you feel, but maybe, a slight hint at what the functional outcome might be. But I would say, at least for my money, we haven't sort of closed the books on how to link that genetic difference to a real phenotypic difference in that dramatic color change.
Yeah, I would agree. I mean, they have no idea how cortex, the protein, does any of this, right? Because its original function has nothing to do with color, with melanin deposition. So I think that's a very small difference. But I would, I don't know if they can do directed modification of these flies, introduce genes into them like you can in Drosophila, right? You can put transposons in and disrupt, and it would be very interesting to disrupt parts of this transposon and see what part is important for this, And
oh yeah, I 100% agree. And that's another, I think this echoes on what we were talking about before. Not only is sort of, genomics opening up all these possibilities for ecological genetics, but so are the actual tools. So if you could go in with a way of modifying the genes and actually put back, in a clean Typica background, just
Right.
that insertion, right? I mean, that's kind of what you really want to do.
That would be great. And then if that worked, then you could start dissecting it and say what part of the transposon, what's the minimum part, and what's it doing, and so forth.
Yeah, no, I agree.
Cool.
Yep.
Cool
I mean, maybe I agree. Maybe one compromise would be, so I think those techniques in a moth that hasn't had a whole lot of experimental attention would be tricky.
Yeah.
But as they point out, there's the cortex gene was first functionally described in Drosophila, so you have the ortholog there.
Mm-hmm.
And so you could use melanogaster and maybe hop in this carb transposable element and see if that has any, like maybe the flies will turn slightly darker. Who knows? That would be kind of a cool.
Yep.
Yeah.
That'd be good to do. Now, people may be wondering, who cares about moths, right? I mean, one part of it, of course, is this whole idea that transposons can cause changes that are selectable and may have important consequences in the wild. It's important to study this in different organisms, and we've gotten a few of them here on TWiEVO so far. This is another example of that, and I think you learn a lot by looking at that sort of transition, don't you?
Oh yeah, you and me both. I mean, I might be exposing a little bit of my bias here, but I really think the transposable elements are the fascinating parts of this interaction in the way that these are sort of, nature's genetic engineers in a sense. But then I would say it's really cool to kind of link that all the way through to then the outcome that is a pretty dramatic one. If you can change the color of a moth, and then that changes the ability of it to be camouflaged, which could be an advantage in some scenario versus another. Now we're starting to build up again back into that biological complexity, and to be able to sort of traverse all the way through from this behavior, which is, can a bird see a moth, all the way to this selfish genetic element that just randomly moved in a different location.
Mm-hmm.
I mean, that to me is just so fascinating and interesting.
And this is a concept we've touched on before. We don't have to have any changes in coding sequence, right? These elements can go in and change the level of an mRNA, of a specific mRNA. You don't have to have a mutation, which could be deleterious. So you can imagine that you could change an amino acid and get black moths, but they would have some other problem because of that amino acid change, because this protein has a role in cell cycle regulation. But here, you don't have to change an amino acid. You just pop in a transposable element that somehow alters the regulation of the mRNAs, and that we've seen in let's see, what was one of the examples where we saw a transposable? Was it the pigeon was a transposable element before, or was it?
Yeah, no,
it was enhancers, right?
So this idea of enhancer shuffling, we saw that it's kind of close by. It's in the butterfly wing pattern.
That's right.
But these things, they're coming up all over the place. We're, in fact, starting to see this in some of our work in my lab. And so stay tuned. I don't want to give away any punchlines, but I'm becoming increasingly convinced that it's really important to keep an eye on these selfish genes. I also have to say, I mean, just from a kind of almost a philosophical standpoint with this story and kind of backing out even farther, I mean, I really think it's interesting how you have, again, at the molecular level, this selfish gene just sort of moving around. But then you can, I think you could draw, this is a little bit of a stretch, but you can draw this idea that it's sort of layering on top of the selfish behavior of humans. So here we are burning tons of coal during the Industrial Revolution, which is changing the environment in a dramatic way, which then allows the selfish gene to propagate in a new way. And so again, there's sort of this weird back and forth.
Yeah, right. Yeah,
all of these collisions between different behaviors at both whatever molecular level, and then also at the societal level about the choices we're making about what we're doing with natural resources.
Yeah, we have all kinds of effects on species out there, right? But there are also natural effects that don't involve humans, right?
Oh yeah,
now, this just happens to be a classic one.
That's right. Yeah,
people study, but I also think that it's important to look at these basic systems, and you learn new things, and then eventually people look at them in mammals and say, hey, the same thing happens here, and we can learn a lot from that, obviously.
Yeah. Yep. Yeah, and we've already seen that in some cases. So if we move into virology, for example, and the idea of restriction factors, right, that are blocking things like the ability of the HIV virus to move around, to replicate. So there are examples of these TRIM genes that you've discussed a lot on TWiEVO.
Right.
And a really interesting case of these trim CYP genes, which are the product of a selfish gene hopping, in this case, a cyclophilin exon next to the open reading frame of a trim gene, and then sort of giving birth to a new immune function, in this case, a new restriction factor, in this case, indirect, but it's the indirect activity of a retrotransposon that's moving around host transcripts and then sort of rearranging them into new combinations.
Mm-hmm.
And so I think that's another, just an example of what you're saying, that the things that are happening in one system kind of happens everywhere.
Yep. Yeah, it is a great paper. I love it.
I do too. Really fun.
It's a lot of fun, and just goes to show this whole idea of these, you can think of them as DNA's hopping around the same way that you can think of mutations arising randomly. They just crop up here and there, and this one popped into a locus, and we may never have seen it, except that it conferred black to the moth and allowed it to evade the bird.
Yeah.
It's just beautiful.
Yeah, that's really fun. And I think, just to highlight again the 2 points, to keep an eye on the role of transposable elements, selfish genes, and the impacts they can have. And then also to echo on your point, which is that not only should we be looking at coding regions, but also how things are regulated. And we're just slowly building up more and more examples of both.
Yep.
And yeah.
All right. This episode of TWiEVO is brought to you by CuriosityStream. This is a subscription streaming service. It's the world's first ad-free nonfiction streaming service. Nonfiction. Okay, it's real stuff. They have over 1,400 titles, 600 hours of content, and it was founded by John Hendricks of Discovery Communications, which is a channel that deals in reality science and not reality TV you can find elsewhere. And you can play these videos on many platforms, the web, the Roku, which I love. It's a great name because it's 6 in Japanese. Ich ni sonchi go Roku number 6.
There you go. Your vacation is paying off.
There you go. Turns out that was the 6th company formed by the guy who formed it, so he called it Roku.
Oh, interesting. Didn't know that. Huh?
Android, iOS, Chromecast, Amazon Fire, Kindle, and Apple TV, 196 countries. And what they have is a wide variety of science and technology content. They also have nature, history, many more topics. They have things like Next World with Michio Kaku. They have viruses, destruction and creation. They actually have a few virus movies in there. One of them is about Zika virus, of course. They have Inside the Human Body, multiple topics relating to the human from creation to survival, the human brain. Microbirth, they look at research that links the way babies are born with their health later in life. Leaps in evolution, a lot of good stuff. And that's why I thought it would be a good fit for all of our podcasts, because there's a lot of good science in there.
Yeah, that's a great fit. And to promote nonfiction in an era where that's maybe not as valued as it always has been is a good, I think it's...
Well, we have an interesting core audience here on Microbe TV Podcast. I think there are people who like nonfiction, right?
Yeah, that's right.
So I think this is a perfect fit. They have monthly and annual plans. They can start at $2.99 a month, which, you think about it, less than a cup of coffee. When you're thinking about subscribing, 1 cup of coffee, and you probably drink 1 a day.
That's right.
So if you never stopped asking why, if you're curious, like we tell you to be at the end of TWiEVO, check it out. Check out curiositystream.com/microbe. Use the promo code microbe during sign-up to get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at startup, at sign up. I always say startup, at sign up. We thank CuriosityStream for their support of TWiEVO. And, we really like the support of ads. We're going to have more in the future because it lets us do different things. So I can tell Nels, hey, I'm doing a podcast here in this city. Why don't you join me? I'll pay your way, stuff like that. I'm really looking forward to being able to do that.
I agree. Really fun to just kind of promote these collisions, and who knows what will come out of it as people and ideas kind of filter together and collide in new and interesting ways. And I have to say, sometimes nonfiction can be better than fiction, the deeper you look.
Yeah, so what is it they say? Some science you just can't believe it's real. It's just so amazing. And that's what we talk about on these podcasts.
Yeah, that's right.
Now, we have a bunch of emails. Want to read a few of them, Nels?
Sure, we should do that.
Let's. First one is from Bill, who writes, Dear TWiEVO folks, it's a beautiful spring day here in Berkeley, California, 72 Fahrenheit, 24 C, 35% humidity, cooling to a more typical 65 S, 18 C later in the week with a chance of rain in a few days. It might be interesting to do a TWiEVO on the evolution of life from beginning to formation of multicellular organisms. This thought was sparked by the following article, which talks about life at a time when nothing modern could have lived. Well, maybe that is not strictly true, as the anaerobes that live in deep rock pores presumably could have lived. That's a fascinating topic in its own right. And he sends a link to that, an article about anaerobes that live in deep rock pockets, but also the main point is an article in Science News. Bubbles may have sheltered Earth's early life. So this is an article in Science News, and it's all about bubbles, pockets of gas trapped on ancient shorelines. Oops, I'm getting a pop-up ad here. Come on.
Well, and then he links it all the way through to the underlying scientific publication from geology.
That's right.
Yep.
Evidence for cavity-dwelling microbial life in 3.22 now, GA tidal deposits. What's GA stand for again?
I don't know. Is that billions of years, maybe?
Maybe, yeah.
Ago? Yep.
So he writes an abstract. Pockets of gas trapped along ancient shorelines gave microbes a cozy place to call home about 3.2 billion years ago. Such a snug hideout could have shielded microbes from UV radiation, not only on Earth, but perhaps on Mars. Earth was a tough place to live on a few billion years ago. No oxygen, no ozone, no protection from UV, says the author of this article, who's a geobiologist. Still, microbes found a way to survive in many places around the world. They glued sand and cells together, forming slimy carpet-like biofilms or mat underwater. Iron dissolved in the water could have screened out radiation, and during low tide, surface microbes might get zapped by UV and die, but communities living below them may have thrived.
Yeah, some really interesting ideas. And I think we've talked a little bit about some of these origin ideas, the really deep evolution. We're talking about, in this proposal, to go from the evolution of life from beginning to formation of multi-cell organisms. Yeah. We're talking, that's a long time. That's billions of years, and a lot of things happening a really long time ago.
You bet.
Super fascinating. Yep.
Why don't you take the next one, which is from the same Bill, I believe.
Yeah, it looks like Bill's a regular in our TWiEVO mailbag here. This is great. Yeah, I really appreciate it. And some great ideas. So let's see, we're getting an update on Berkeley weather. We've gone down a couple degrees to 67 Fahrenheit, 19 C, light breeze, bit more humid than normal. Some rain expected tomorrow. I'm sure you have or will see, but here it is anyway. The most characteristic feature of Darwin's finches is the diversification of beak morphology that has allowed these species to expand their utilization of food resources in the Galapagos archipelago. A team of scientists from Uppsala University and Princeton University has now identified a gene that explains variation in beak size within and among species. The gene contributed to a rapid shift in beak size of the medium ground finch following a severe drought. The study is published in Science. And then we have a link as well as some...
It's not a transposable element, is it?
It's a good question. I remember this paper. That would be great, right? I mean, it's a very similar topic in the sense that you have this iconic, example of evolution that we sort of celebrate the history of, the same way we do with the peppered moths. A really dramatic morphological difference, in this case, the beak morphology. Were any of these guys eating moths? If we could combine those two things, we would really be in business. We could collide them.
So they have this gene called HMGA2, which varies among finches with different beak sizes.
Okay, yeah.
I don't know anything about that.
I remember seeing the paper, but I haven't really dug in on it. Another great example. I think that's not a transposable element. So they aren't. And maybe that helps us balance our arguments a little bit. it's not the selfish genes that are doing everything, just a lot of things.
Well, they say that we discovered a genomic region that includes this HMG gene. So who knows? Maybe there's more to it than just the gene. I don't know if they identified any polymorphisms as was done in our paper. Right? Yeah,
it's a good question. I'm not sure the genetic resolution that they're down to yet in that paper. But it just, again, illustrating that people are starting to put these things together, though, in new ways, and to really start to zoom in on the genetic basis of a lot of really cool, and in this case, another iconic trait.
Yeah, I'm going to find out. So two loci have major effects on beak morphology. One of them is a transcription factor gene called ALX1, which is only 7.5 megabases from the HMG-A2 gene, which is implicated in this study.
I see. Yep. Yeah, and the nice thing about some of these studies is we're also, in addition to, if you come at it from the functional side, we're starting to learn about how transcriptional regulation works, how near or far away loci are that sort of influence each other in terms of transcriptional regulation.
So this is really the same theme as today's
I agree.
episode for sure, right? They're getting under the basis of the variation. That's the basis for selection.
Yeah, and in this case, they say, the environmental change is not a bunch of industrial coal burning, but a severe drought. And they think that's what they're saying, potentially selected on this shift in beak size. But again, a really strong environmental shift that then can open up the space for natural selection to really act. These perturbations, whether it's broadly defined, we see these really dramatic environmental differences. Yeah.
Our next one is from Anthony, who writes, the smallest avian genomes are found in hummingbirds, and links to a paper in the Royal Society where it says, the abstract, it's often been suggested that the genome sizes of birds are constrained relative to other tetrapods owing to high metabolic demands of powered flight and the link between nuclear DNA content and red blood cells. This hypothesis predicts that hummingbirds, which engage in energy-intensive hovering flight, will display especially constrained genomes. So they looked at genome sizes for 37 species of hummingbirds, and it confirms the prediction. Smallest genomes.
Yeah, I think this is jogging my memory. So thanks, Anthony. I think I forget the episode, Vincent, but we were having a conversation about genome size. And I think I just sort of offhandedly suggested this. It must have just been sticking in my mind, that I was kind of joking, but I guess I did see this. So the hummingbird genomes are small. I think if you go on in that abstract, they are careful not to put too much of I think there's an important lesson in here, not to, so it's easy to think, oh, well, the hummingbirds themselves are really small or have this high metabolism, so maybe they have to economize their genomes. But then they point out that the genome size was reduced before the divergence of extant hummingbirds. And that, in fact, I think genome size appears to have increased in 4 unrelated hummingbird species. And so that is a nice example not to read too much into some of the genetic variation that we see, right?
Yep, absolutely. Yeah. All right, dear, Bill's up. Bill's back.
All right, he's really filling the mailbag this episode. I love it.
That's your turn.
Oh, okay. Dear TWiEVOers, the most recent TWiEVO, apparently credible DNA has been extracted from ancient horses preserved in permanent frost. In June 2013, a group of researchers announced that they had sequenced the DNA from a 560 to 780,000 year old horse using material extracted from a leg bone found buried in permafrost in Canada's Yukon territory. In 2013, a German team reconstructed the mitochondrial genome of Ursus deningeri, another ancient horse species, I guess, more than 300,000 years old, proving that authentic ancient DNA can be preserved for hundreds of thousands of years outside of permafrost.
It's interesting. We must have been talking about that, right?
Yeah, so some of the Neanderthal, right?
That's right.
And how far back you can go.
Yeah.
And so this is a nice suggestion, right, or a nice illustration that perhaps there's, and I think this is already happening, but people are really It's kind of like gold mining almost. Looking for these ancient remains and trying to pan out genomic material from them as far back as you can go. With the idea that's probably, the farther back you go, in some ways it's adds value to being able to really not have to infer what happened during evolutionary time or during our history, but to actually not just to glimpse it, to see it and how it 100% was. If you can, with some caveats being that it's really hard to, it's a technical challenge to distinguish between contamination and real ancient DNA. And also the farther back you go, the more chances there are for degradation. So it becomes harder to reassemble these things into long annotated genomes. But it really is exciting.
Yeah, it's great stuff. I love it.
Yeah, me too.
Next one's from Sophie. Dear TWiEVO's Dr. Racaniello and Dr. Elde, thank you for another great podcast celebrating science and the wonder of nature. I'm an MD living in Denmark, slaving through my PhD in epidemiology, so not much of a lab person, unfortunately. You don't have to read this email or post it anywhere. I just want to suggest a researcher you might want to try to bring on the show someday, if possible. I'm a big fan of Professor Eski Willerslev. He's published a bunch in Science and Nature on evolution. He is, on top of a great researcher, fantastic at communicating his results and very down to earth. I know, like you and every other accomplished researcher, he's extremely busy, but if one of you should ever happen to come to Denmark or he should come to the States, you should consider contacting him. He doesn't know much about viruses, but I know he is interested in trying to sequence ancient microbiomes, bacteria, virus, fungi of ancient people. He has lately published data where he found Yersinia pestis DNA in samples from Bronze Age Homo sapiens. But Eski is really just a great guy to talk to about the evolution of Homo sapiens and how to sequence ancient DNA. Hey, Nels, you want to go to Denmark?
Yeah, I do. That sounds perfect. Great science, great cuisine. Some of the great. Go in the summer. I think you're still far enough north that you get some sunlight. It's a good idea. Thank you for this email, Sophie. These are all great ideas. Working on you have an MD, working on a PhD in epidemiology. There's always a chance for a third act, which is to do some lab work, if that ever moves you, I would say.
Of course, you can do it. Yeah.
I've come a little bit across Eska Willerslev's work before. There's actually, I think, a really nice profile of him in the New York Times not too long ago that someone came across. But yeah, I hope that our paths collide sometime. Really interesting work along the lines that we've been discussing with ancient genomes. And then what I really like are these new ties to the ancient microbiomes, as well, as people are not only kind of, trying to get the DNA from the bone of a Neanderthal or a Denisovian, but also saying, oh, wait a minute, what about the passengers that have been there this whole time? The Yersinia pestis stories are really interesting. Actually, this fellow, Wyndham Latham, who's at Northwestern, I think his lab's about to relocate to the Pasteur Institute.
Wow, really?
Yep. But he's been doing, or thinking about the evolution of pestis using some of these data sets and then actually doing some of the microbiology, and then trying to link the genetic differences to the key sort of pathological differences between pestis, which causes the plague, versus things like pseudopestis, which has a much more mild impact on human disease.
I had Wyndham on TWiM a couple of episodes ago.
Oh, yeah. Yep.
Talked about that and finding pestis DNA. They were sequencing teeth of old buried humans, and they had this dark DNA. They couldn't figure out what it was. And it turned out to be Y. pestis.
Yeah, and in fact, he might have been, I think he was mentioning to me that it was a Danish group. And so I wonder if that's
maybe it was him.
Yeah, it might be the same crew. And so a great example, I think, of what can happen, so Wyndham, in this case, is coming at it from a traditional microbiology perspective in all of those, but sort of open to the ideas and kind of learning on the fly how to think about some of the evolutionary ideas. And so I give him a lot of credit for branching out and sort of meeting evolutionists halfway, so to speak, breaking down some of these barriers. It can be at times, a little intimidating, frustrating. It can happen in fits and starts, but it's the insights and the fun of doing that work, I think is really
well. We'll keep our eye out, and if he's ever here or we're over there, we'll hook up.
Yeah, good idea.
Let's see. Let's do a couple more. What are we up to, Robert? I think your turn with Robert now.
Okay, so I haven't read the paper yet, but saw this come by and thought of you guys, especially after listening to the Pigeon podcast. Maybe this is run-of-the-mill evolutionary sequencing work, but it's pretty darn cool to an engineer like me. And then we have a link to a Nature Communications paper. The origins of giraffes' imposing stature and associated cardiovascular adaptations are unknown. Okapi, which lacks these unique features, is giraffe's closest relative and provides a useful comparison to identify genetic variation underlying giraffe's long neck and cardiovascular system. The genomes of giraffe and okapi were sequenced, and though comparative analyses of genes and pathways were identified that exhibit unique genetic changes and likely contribute to giraffe's unique features. Some of these are in the Hox, Notch, and FGF signaling pathways, which regulate both cytoskeletal and cardiovascular development, suggesting that giraffe's stature and cardiovascular adaptations evolved in parallel through changes in a small number of genes. Mitochondrial metabolism and volatile fatty acid transport genes are also evolutionarily diverged in giraffe and may be related to its unusual diet that includes toxic plants. Unexpectedly, substantial evolutionary changes have occurred in giraffe and okapi in double-strand break repair and centrosome functions. So first of all, Robert, thank you for lifting up this paper. I agree, it does sound really interesting. A couple of points I guess I would bring up kind of right out of the gates, which again, it kind of echoes on what we've been talking about today, is, as you're picking a system to map how genetic divergence has happened in sort of interesting ways, you always want to consider the resolution. So in today's example with the moths, that divergence we know happened over the course of a couple hundred years, really recently, right? The moth is the same species; it just changed color, and so you have this sort of tight resolution or a defined difference. I think the giraffe okapi comparison is super fascinating. From an experimentalist standpoint, it feels also really ambitious because here you have a last common ancestor that goes back, I don't know the exact numbers, but I would guess at least, on the order of 1 million or more years. There's a lot more time for a lot more things to happen. And then as Robert points out, you have all of these really interesting differences, the cardiovascular differences, obviously the just the length of the neck and the developmental biology that goes into that. and then there's sort of, I haven't read the paper, but I get the impression at least that, they're starting to then map all of these genetic differences. And again, it's really tricky, I think, at this stage to kind of put your thumb on it and say, oh yeah, this difference in Hox signaling has this outcome. And so I think what we're seeing here is super fascinating biology. From an experimental standpoint, much harder, in fact, to get that high resolution view that we focused on today. Yep.
Yeah, unless there were just one nucleotide difference between the two, it could be pretty hard to say this is it. Yeah, but nevertheless, case of macroevolution. So we were talking about microevolution today. Yeah. These massive changes, and that's a frontier, the wilderness of evolutionary biology and genetics. All right, let's take one more from Anthony. He writes, I only listened to the recent TWiEVO once, so perhaps I heard wrong. I thought that you said that parasitic wasps deposit larvae. They lay eggs, as may be seen in the image at TWiV 179. And I think this has come up before right now.
That's right.
We made a mistake.
This was, I think, my Freudian slip on eggs versus larvae. Yeah.
By the way, the parasitic wasps were a focus of Darwin's. Quote, I cannot persuade myself that a beneficent and omnipotent God would have designedly created the Ichneumonidae with the express intention of their feeding within the living bodies of caterpillars, or that a cat should play with mice. Indeed, Chuck.
That's right. Yeah, all of these connections back. We keep seeing it again and again. The pigeons, the parasitic wasps, all of those sort of dramatic examples of evolutionary diversification, all this complexity of behaviors and forms, which sort of gave birth to the field, keeps reverberating up and will hopefully inspire this and future generations to do kind of as dramatically interesting things.
Yep.
Yeah.
All right, let's do some picks, Nels.
Yeah, great idea. So I'm going to go something a little out there, I guess, but I guess connecting in some ways loosely to popular culture and ecological genetics. So this is this new craze that's just come out of nowhere, this Pokemon Go game. Have you?
Oh, yeah.
You come across this? Yep.
You bet.
So I found a short blog post or a short article on the topic, is Pokemon Go good for science? And so on the one hand, we have this kind of crazy computer game with these Japanese anime-like creatures that people are chasing all over their neighborhoods and kind of getting in trouble. Wasn't there a crew in Central Park that kind of went crazy the other night or something like that? I think that was in the
I didn't
news.
hear about that one, but I know there have been some people. The thing is, you set a point where people will come and collect Pokemon, and then you can rob them and do bad things,
Yeah, sure. So people are putting out these lures, I guess, and the idea is as part of the game, you're taking your phone and kind of pointing the camera in different directions and hoping to come across one of these mythical anime characters that then you get points, and I don't know the details, to be honest. But so some, ecologists and naturalists have pointed out that, you can go out and cause all this excitement to chase after these mythical things. And there's, 150 species of them, fake species of them. Whereas what about real nature? Are we forgetting the millions of beautiful and incredible animals and other forms that are out there, and sort of not maybe putting our focus in a funny place?
Yeah, it's an interesting point.
It's an interesting point. I think for me, at least, I think it speaks to a deeper kind of impulse of our species, of our psychology, which is there's just this natural fascination with some of this. And so this could be something that we could tap into as scientists, and just across the board in terms of just the curiosity of people and anything, you could spin it to the idea that this could actually sort of be a new motivation for the next generation of field ecologists, if maybe they have their interests sort of born out of this game, but then actually extend that into real life. But who knows?
It's an interesting point. So we were in Japan when Pokemon Go was released, and my younger son, who we brought, is a big anime fan, and I said, don't you want to play this? And he said, well, it hasn't been released in Japan. Can you imagine? The whole Pokemon thing originated in Japan. It's only been released in the U.S. and then parts of Europe. So when he got home, he immediately started playing it, and he says, I'm going for a walk, and he never does that. He never goes for a walk. And he was going to one of these places where someone has set I don't know I forgot what you call it, when you set a place where then people go and look.
Yeah, I don't know. Yeah. But that sounds right. Yep.
So what I do is when I have an Instagram channel where I post sciencey stuff, photos, and when I travel, I take photos of weird creatures that I see.
Cool.
All right, so I found some interest. I found a walking stick in Japan, an interesting bug. I posted this bug, and I said, I don't know what this is. And someone wrote, that is an ash borer, and it's an invasive species that destroys ash trees. And I didn't know this. I found a bug on the beach, and I put it up. And so I think this is cool. I think there's a lot of stuff out there. And when you're on vacation, you have time where you can take a look at stuff and take pictures. So I like to do that. And by the way, Instagram.com/PROFERR, if you want to follow me. When I'm in the lab, I post a lot of lab-like pictures and sciencey stuff, but when I travel, I try and look at biology in a broad way and put the pictures. So it's the same idea as this article is saying, get out there and look at what's around.
Yeah, this is Racaniello Go.
That's right, Racaniello Go.
Yeah, I have a friend who just posted on Facebook. He got one of these camera traps. It's like wildlife ecologists will set up to monitor how many species are moving through. Just set this up in his backyard in Palo Alto and started taking pictures of all of these rodents that were coming through. I think there's a jackrabbit that came through.
Cool.
And just all this, some voles and just all kinds of stuff. And so I'm kind of tempted to do that. That will be as close as I get, I think, to Pokemon Go would be to get one of these camera traps, because there's a raccoon and I think some other critters in my backyard that I'd like to get a handle on that come out at the middle of the night, but I never see them.
Well, we live in a suburban area. We now have foxes and coyotes walking around in the backyard.
Oh, yeah. Cool. Yeah, it's great.
My pick is a book called 7 Brief Lessons in Physics. The author is Carlo Rovelli. And it's just a wonderful example of good science writing. And here he picks 7 discoveries in physics and explains them in a way that's really understandable, but are correct. And, I just stumbled on this and I started reading it and I loved it. The first chapter is about Einstein and relativity. It's called The Most Beautiful of Theories. And I just want to read you the first paragraph because it's lovely.
Please.
In his youth, Albert Einstein spent a year loafing aimlessly. You don't get anywhere by not wasting time, something, unfortunately, that the parents of teenagers tend frequently to forget. He was in Pavia, it's in Italy. He had joined his family, having abandoned his studies in Germany, unable to endure the rigors of his high school there. It was the beginning of the 20th century, and in Italy, the beginning of its industrial revolution. His father, an engineer, was installing the first electricity-generating power plants in the Paduan Plains. Albert was reading Kant and attending occasional lectures at the University of Pavia for pleasure, without being registered there or having to think about exams. It is thus that serious scientists are made. I just think it's beautiful.
Spectacular.
You have to waste time sometimes, just waste time. You can be thinking about stuff, and that's how you develop. So people who have kids, don't worry about them wasting time. It'll be okay.
I agree.
So that's the theme of the tone of this book. It's really lovely.
That's spectacular. Great pick.
So you should check it out. It's a tiny book you can read in no time, and I think
Very
it's
cool.
really well done. I'm always on the lookout for good science writing, and that's a good one.
Yeah, it's clear even from that first paragraph. And
Wonderful.
I don't know that aimless loafing. I don't know if I'm going to be able to fit that into specific aim 1, but I would love to do that.
Yeah, isn't it too bad? Well, you're beyond the part of your life when you should loaf now. Unfortunately, that's a formative time. It's teenage. You're past the teen. You should. Didn't you loaf ever in your life?
Oh yeah, I do quite a bit of loafing stuff.
Well, that's why I think it's important to take a little time off, right, and go loaf somewhere because
could not agree more.
You discover different things. You get a new perspective. It's really good. You don't have to work every day of the year. You can take time off.
Recharge the batteries.
And so, well, that's TWiEVO 10. Nels, we made it to 10.
Double digits.
Double-digit TWiEVO. Looks like we're going to be here for the long run.
Yes.
You think so?
I do.
All right.
We'll see if it will be measured in billions of years by the time it's all said and done.
That's right. Well, after I'm gone, you have to keep going with TWiEVO. I'll teach you how to do it. Get yourself another. I mean, this is not happening tomorrow, but
no.
20 or 30 years, you should do it. I'd love to figure out a way to keep all these podcasts going. That's one of my goals for the next decade, I guess.
Yeah, definitely.
Anyway, TWiEVO is at iTunes. It's at microbe.tv/TWiEVO, many other places. If you have a podcatcher on your iPhone or Android device, or, it's a program that grabs podcasts, you can get them anywhere. And you can hear TWiEVO there as well. And if you'd like to help us, we've been doing ads, but we're also looking for listener support. You go to microbe.tv/contribute. There are a couple of ways that you can help us. One of them is at Patreon, where you can be a patron of the podcast arts, and you can contribute as little as $1 monthly, and or you can give us funds other ways. They're all at microbe.tv/contribute. And as I said, we want to use it to be able to travel more and do different things with the podcast that we haven't been able to do, get more creative as well. And of course, don't forget to send us your questions and comments. We love them. TWiEVO at microbe.tv. You can find Nels at cellvolution.org. He's also on Twitter, Earlybird, ELEARLY Bird. Thank you, Nels.
Thank you, Vincent. Great as always to be with you.
Good to have you back again, and our monthly TWiEVO is in the can. I am Vincent Racaniello, by the way, and you can find me at my blog, virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.